Good evening, everyone. Thank you so much for joining us tonight. My name is Kate Bruns, and on behalf of Harvard Bookstore, the Harvard University Division of Science, and the Cabot Science Library, I am very pleased to welcome you to our program tonight with Venki Ramakrishnan discussing his latest book, Gene Machine: The Race to Decipher the Secrets of the Ribosome. In just a moment, I'm going to turn you over to Professor Stephen Harrison of the Department of Biological Chemistry and Molecular Pharmacology at Harvard Medical School, who's going to introduce the program this evening. But first, I'd just like to take a quick moment to tell you about this Science Book Talks series, a collaboration between the Harvard Division of Science, the Cabot Science Library, and Harvard Bookstore. The series features talks throughout the academic year by the authors of recently published books on a wide variety of science-related topics. Next up, on June 5th, we will welcome the acclaimed author of The Strangest Man, Dr. Graham Farmiloe, for a discussion of his latest book, The Universe Speaks in Numbers: How Modern Math Reveals Nature's Deepest Secrets. He will be joined in conversation by Harvard Physics' very own Jacob Berendis. To learn more about the Science Book Talks and to learn about Harvard Bookstore's many other upcoming events, you can visit us online at harvard.com and sign up for our weekly email newsletter. Tonight's talk is going to be followed by some time for your questions, after which we will have a book signing and refreshments in the Cabot Science Library, just outside this room and across the Science Center entryway. And now I will turn things over to Professor Stephen Harrison.
So as the size of the audience suggests, and as the topic of the book, which is really in part at least an autobiographical memoir, indicates, I won't spend any time introducing Venki Ramakrishnan at all, except to comment for 30 seconds on the subtitle, The Race to Decipher the Secrets of the Ribosome. Scientific races, and you've probably read or heard about some of them, like any race, tend to have a defined goal line, but there are lots of ways of approaching that goal line, or even in fact, in defining it in truly scientific terms. And so some people get there, if you wish, by being very good at organizing the right kind of team, and other people by knowing very deeply what the right kind of question to ask is, and then figuring out how to get there. Venki is certainly in the latter category, a very deep and very fine scholar, and that's characterized, I believe, specially his role in this race to decipher things. He's also a very felicitous prose stylist, as those of you who have read, are reading, or will read his book, which I heartily recommend. And he's also now, unusual among scientists, taken on a serious task in public service as president of the Royal Society. Well, you've come here to hear him, so Venki.
Thank you very much, Steve, for that very kind introduction. So I'm going to start off. I want to try and finish on time, so I'm going to not waste too much time. But I will say I've noticed a lot of people who I know are biologists in the audience, and my lecture is really not meant for biologists, it's meant for the general public. But I take heart from Enrico Fermi, who said, never underestimate the pleasure you give people by telling them what they already know. So without further ado, I will plunge into the book. Now, this book has 4 themes. One is about the ribosome itself, which is this amazingly complex and ancient machine that reads the code of life to make you and me and every other life form. The other is a more personal story, which is interwoven, which is my life as an outsider and what it suddenly felt like to suddenly be in the middle of a high-stakes race or even in the pursuit of an important problem. A third is about the human messiness of science. Often science books, there are some notable exceptions, including from a former Harvard faculty member, but by and large, science books sanitize what science is like. In hindsight, everything is logical, everybody's altruistic, it's a noble pursuit for the truth, etc. But in reality, science is done by humans with all of the problems that humans have: egos, personalities, rivalry, question of competition versus collaboration, and how prizes throw a sort of combustible element into this already somewhat complicated mix. And then finally, I might end with one slide on lessons from my own life in science, which are far too late for me to learn from, but which perhaps young people in the audience might find something from. So I want to start off by asking the question of what is a gene? If you ask if you walk down outside, even around Harvard Square, and ask people what is a ribosome, most people won't have the slightest idea what a ribosome is. If you ask them what is a gene, most people will say, oh yeah, I know what a gene is. It's that thing that gives us, traits, our characters. We have good genes and bad genes that we inherit from parents and so on. But if you really ask them, well, what is a gene? They won't be able to tell you, okay? And they'll be particularly surprised to find that humans have actually, this is an overestimate from 2001, but we have only about 25,000 genes. And we don't have any more genes than a weed called Arabidopsis thaliana or a worm called C. elegans. So anytime we think we're so much more at the pinnacle of life, we should learn some humility from this fact. And the reality is, life does not select for intelligence. It selects for survivability. And intelligence is a trait that helped us survive in certain circumstances. It may, in fact, lead to our extinction, for all we know. But anyway, so we have thousands of genes, and even bacteria have thousands of genes. And so the question is, what are genes? And what genes are essentially long chains made up of building blocks called amino acids. And there are about 20 types of amino acids. So you could, in principle, represent a protein chain by a string of letters. Now, these amino acids, each one of them, they have different properties. Some of them don't like water, some of them are charged, some of them are neutral, and so on. So they're capable of very different chemical properties. And as a result, the order of the amino acids that make up a protein contain in them the information on how to fold up the protein into a particular three-dimensional structure. Now, pause and think about how remarkable this is. It would be as if I took several strips of paper and wrote down a different sentence of different lengths on each strip of paper. And depending on what I wrote, the strip of paper magically folded itself up into a particular three-dimensional shape. That is how amazing it is, except it's not a miracle, it's chemistry. It has to do with the way that amino acids interact with each other. And just to give you an example of the variety of shapes and functions proteins can have, here is a protein which is a long filament-like protein which consists of 3 strands that are woven around each other, and it's called collagen, and it's what makes up your skin and cartilage and connective tissue. In fact, it's the most abundant protein by weight in us. This is a protein called hemoglobin, which captures oxygen in our lungs and transports the oxygen through our blood to the tissues and muscles where it's needed. And this is a protein called rhodopsin, which is present in the membrane of cells in your retina and detects light and allows you to see. So these are just 3 examples from the thousands of proteins that we have. And these proteins carry out all of those thousands of functions. Now, you might ask, well, okay, genes contain information on how to make proteins. I've just told you what proteins are like. How do genes actually contain that information, and how does that work? Well, genes reside, with some exceptions like viruses, some viruses, it resides in a molecule which is familiar to all of you, which is DNA. And DNA, as you know, consists of 2 strands. And instead of being made of 20 types of building blocks, DNA is made of 4 types of building blocks called bases. And the remarkable thing about the DNA structure was that the structure showed that each of the 4 base pairs has a particular partner among the other 3 that it pairs with. So A will only pair with a T, and C will only pair with a G, and vice versa. And what this meant is that if you have a particular order of bases on one strand, that would specify the order of bases on the other strand because of the way they would pair up. And so if you separated the strands, each one contained the information you needed to make the other strand. And this is how the problem of centuries-old problem of how do you get 2 cells, or 2 of anything, from one was solved in molecular terms. But what the structure didn't tell you was how you could somehow, from the order of the 4 bases, figure out how to make a protein, which consists of 20 types of amino acids. And this took almost a decade after the structure was solved, actually a little over a decade. And it turns out that what happens is, in a long DNA molecule, different stretches represent genes that code for different proteins. So, for example, we have 23 chromosomes, and we have about 20,000 genes. So you could see that each chromosome on average might contain 1,000 genes. And so they're sort of spread out over different parts of the molecule. And what the cell does is, now if you were to go to the Library of Congress or where I live to the British Library and say, I want to read a particular book, they're not going to let you read that book. It's too valuable. What they'll say is, we'll give you a copy of the book to read, and you can take it with you and you can read it. And the cell does something like that, in that you can think of DNA and our genome as the library of all of the information, of genetic information. And then it makes a copy of each gene as it needs it in order to be read to make a particular protein. And that copy is a very similar molecule to DNA called RNA. And then, which also consists of 4 types of bases, and the order of bases is exactly what it would be like on one of the strands of DNA which contains genetic information. Somehow from there, you have to get to this totally different type of chain, which consists of 20 types of amino acids. Now, of course, if each base specified an amino acid, you could only code for 4 amino acids. And even if you read them 2 at a time, you could code for 4 times 4 or 16. So not surprisingly, nature reads these bases 3 at a time, and they're called units called codons, because it's the unit of the genetic code. And very early on, Francis Crick realized that amino acids wouldn't be able to recognize these codons directly. So there must be some sort of adapter molecule. And that adapter molecule is another kind of RNA molecule called tRNA, or T is for transfer, because it transfers amino acids to a growing protein chain. And at one end, it has 3 bases that read the 3 bases by matching, by pairing in the gene. And at the other end, it brings along the appropriate amino acid. And so for a different codon, you would have a different tRNA molecule that would bring in a different amino acid. And somehow these amino acids then have to be stitched together to make a protein. Now, of course, this doesn't happen by itself. It's a highly complicated and ordered process. And the first hint of what might be happening was when cell biologists, notably George Palade, showed that newly made proteins occurred in these blobs in these organelles called endoplasmic reticulum in our cells. And when these blobs were isolated, it was found out that they were all about the same size, but if you look closely, there's a divider in each particle, And what it meant was that each particle actually consisted of two pieces called a large and a small subunit. And these particles were isolated from a fragment of the cell. But you break open the cell and you just fractionate different parts of the cell. And the fraction from which these particles were isolated was called a microsome, which are really fragments of the endoplasmic reticulum. And when they analyzed these particles, they found out they were about 2/3 RNA, which stands for ribonucleic acid, and 1/3 protein. So they were originally called the ribonucleoprotein particles of the microsomal fraction. Now, of course, if you wanted to talk about it, this was rather inconvenient. And so Howard Dintzis at some meeting in the 50s said, look, why don't we just call it the ribosome? And that's the name that's stuck ever since. People went on to analyze the ribosome, and they found out it was incredibly complex. Even in bacteria, which have the simpler version, it has about 50 proteins and 3 large pieces of RNA distributed among the 2 subunits that together make up the entire ribosome. And then people figured out, through very clever biochemistry, a worldwide community working for 10 or 20 years, figured out that the ribosome has 3 slots for binding those adapter tRNA molecules. The small subunit binds the mRNA, which contains the genetic information. And the ribosome figures out where to start making the protein, by where to start reading the genetic information to make proteins, and brings along the first tRNA with the first amino acid. And then in the second slot, it selects just the right tRNA that corresponds to the 3 bases on the gene. So it's the gene that's specifying which tRNA comes in and therefore which amino acid is brought to the ribosome. And then the ribosome, if it's the correct one, accepts it and then stitches together a bond between the first and second amino acid. And then the whole assembly moves, the old tRNA is kicked out, and then a third tRNA comes in, and only if it's the right tRNA is it accepted, and the ribosome then stitches a bond between the second and third amino acid. So you see what's happening. It's choosing tRNAs depending on what the code is specifying on the gene, and then at the same time it's stitching together the amino acids brought in by those tRNA molecules. And this is how genetic information is used to build a protein in exactly the right order specified by the information in the gene. Now, all this was known when I first entered the ribosome. Now, the book is organized as a sort of interweaving of science and my own personal story. So I'm using my personal story to give it a narrative structure and dribbling in the science as I go. And so I start off by pointing out that at the age of 19, I graduated with a degree in physics from my hometown university in Baroda, India, and went off to Ohio University in Athens in southeastern Ohio to get a PhD. And this is where I spent the first 5 years of my life. And I should say my most notable accomplishment, perhaps my only notable accomplishment during those 5 years, is that I met and married my wife. And you might ask, well, what happened? Well, here's the problem. I started doing physics. I took all my coursework. I passed my comprehensive exams. All of that was fine. It was no problem. Then I started doing research. And this is what I found. I found it was actually very hard to actually make really original contributions in physics. Okay? And one of the problems is that physics is a very mature discipline. And there'd been very few what I would call really big breakthroughs, even in the few decades, previous few decades when I'd been a graduate student. And I thought, if I stayed on in physics, this is what I would be doing. I would be doing boring calculations that, didn't actually improve understanding, but that might, I could publish, maybe even get tenure possibly, but, it wouldn't actually really be advancing physics. I felt I didn't have that sort of aptitude or talent. And then I looked at biology. I used to read Scientific American, and I realized that every issue of Scientific American in the 70s, and even probably today, contained major breakthroughs in biology. You know, that was a time when DNA was, people had first figured out how to sequence DNA. Some of you may know Wally Gilbert, who was here at Harvard. And you know, the structure of molecules inside cells, even the organization of cells, immunology, all of this stuff was being, you know, broken wide open. And I knew that many physicists had made this transition, including people like Max Delbruck and Max Perutz, Francis Crick. So I knew it was possible. And I had written to a number of people, universities, to ask whether I could go to graduate school again in biology. But you know, at Yale, they said, we can't take you as a graduate student, but some of our faculty might offer you a postdoc. And one of the people who offered me a postdoc, oddly enough, was Tom Stites, with whom I ended up sharing the Nobel Prize. Another was Don Engelman, and I turned them both down, saying, I don't know any biology, and I really need to go to graduate school. And so I went off to University of California, San Diego, which accepted me into their graduate program with the idea that I might do a second PhD. And so I had, by this time I had gotten married. I had a 6-year-old stepdaughter and a 4-week-old son and a very tolerant wife who didn't mind my going back to graduate school with a 4-week-old kid and a 6-year-old stepdaughter in tow. And we packed our stuff in a Ryder truck and, you know, drove off to California, which is where I spent 2 years. And the thing I remember most about biology is that in the first week when professors would talk about their research to sort of introduce new graduate students, I couldn't understand a thing of what they were saying. And I realized that I needed to go back to undergraduate classes. So I spent the first year taking undergraduate biochemistry, cell biology, and genetics. And I had a PhD in physics, and I was competing with all these cutthroat pre-meds. So there we were. So by the end of sometime during my second year, I felt I'd learned enough biology. I'd learned how to do experiments and, you know, do the sort of things that involved biochemical research. And I thought maybe I don't really need to get a second PhD. And around this time, I read another article in Scientific American, and one of the authors was Don Engelman, who had offered me a postdoc. And so I wrote to him and I said, look, you wanted to offer me a postdoc when I didn't know a damn thing in biology, and now I've actually learned some, so maybe you're still interested. And so he put me in touch with his collaborator, Peter Moore, who interviewed me and offered me a postdoc. And so there we were 2 years later, crisscrossing the country again to go to New Haven, where I worked for this man. He has Peter Moore, who has a somewhat glowering expression in this picture, but he's actually a very nice person. And we were trying to understand where the different proteins were in the small subunit of the ribosome. There were about 20 of them. Nobody even knew where they were on the ribosome, and it used a somewhat esoteric technique called neutron scattering. And I like to joke that a third of these balls are mine. But anyway, by the end of this, I applied for a number of faculty jobs, and actually about 50, distributed between research universities and four-year colleges, and I got exactly 0 interviews. And you can sort of see why. You know, here I was working on a problem that was already considered old-fashioned and using a technique that nobody had heard of in a biology department. And you know, I'd had this sort of weird you know, track record. And the four-year colleges probably thought, you know, this guy has this long, unpronounceable name. We don't even know if he can speak English, you know, let alone teach. Okay, so I went into pile B in both cases, and I ended up going to Brookhaven National Lab, where I continued to do this neutron scattering for things. But the real problem was it was sort of a dead end, because it wasn't getting us any closer to understanding how the ribosome would work. And to understand what I mean by that, one important thing in science is that every time you've been able to see things, visualize things at a different level of detail, it has dramatically transformed science. I'll give you just a couple of examples. In physics, the ability to see planets and their moons using a telescope, Galileo, really transformed our view of physics and astronomy. And another example is in biology, the ability to see things that are microscopic. Van Leeuwenhoek and Robert Hooke led to the discovery of microbes and bacteria, and also led to the idea that living things are organized in units called cells. Both of these were completely transformational. The same was true of molecules. If you want to see something, if you want to understand something, you've got to understand what it looks like. And I'd give you the example of a car. If you just look at it from outer space, it'll look just like a dot moving around. You get closer, you start seeing it's got lights and wheels. Still doesn't tell you how a car works. If you open it up, you start understanding how a car might be put together, but you don't understand how it works. It's only when you know the details of what a car actually looks like, how an engine is put together, how it's connected through a crankshaft, et cetera, that you understand how a car might work. And it's the same with molecules. Now, if you want to look at small objects, what you would normally do is you would take an object and have a magnifying glass or a microscope, and you would then obtain a magnified image, and that would allow you to see detail that you couldn't see with the naked eye. Now, what a lens does is it recombines scattered rays from the object and then produces an image by recombining them. And depending on the geometry, the image can be much larger, and so you can see detail. But the thing you have to keep in mind is the lens does not produce the information. It's simply using the information. So in other words, if there were no lens here, the scattered rays here would still have all of the information you needed to make that image. The lens is simply recombining them. And now, if you wanted to look at molecules, though, you couldn't do it this way. And the reason is that the distance between atoms in a molecule is several thousand times smaller than the wavelength of light. There's a very old law in physics from the 19th century that you can't distinguish two objects that are closer together than about half the wavelength of the light you're using. So how would you look at molecules? Well, this was discovered by Lawrence Bragg, who did this when he was a graduate student and went on to become the youngest ever Nobel laureate in science, still is the youngest Nobel laureate in science at the age of 25. And that is, you can take molecules and persuade them to stack up in very regular three-dimensional arrays. And what they then form are what we call crystals. That's what crystals are. They're regular three-dimensional arrays of something, of a molecular object. And so here's a picture of crystals. And what you would then do is hit them with X-rays. Now, X-rays, it turns out, do have the right sort of wavelength to be able to see detail. But there aren't any good lenses to focus X-rays and produce an image as you would with light. And even if there were, by the time you hit it with enough X-rays to see an image, you would end up frying the molecule. So the way you do it is you hit crystals with X-rays, and that amplifies the signal from millions of molecules which are all sitting in the same orientation. And then you measure the scattered rays. And this is what the scattered rays would look like. Because it's a crystal, the rays are scattered along certain directions, and I try to explain that in the book. And then you would do in a computer what a lens actually does, which is you would recombine these rays, the information in these rays, to calculate the three-dimensional image of the molecule. Okay? And that's how this technique called crystallography works. And that's how, for over 100 years, molecular structures have been directly visualized. Now, I didn't know any crystallography, and I was coming up for tenure at Brookhaven National Lab, and they asked me, if we give you tenure, what will you do? And I said, well, if you give me tenure, the first thing I'll do is stop what I'm doing, which seems to be a bit of a dead end, and go on sabbatical to learn crystallography. So they were somewhat taken aback by my honesty, but to their credit, they let me do that and gave me tenure. Okay, and so I decided to go to a very famous lab called the MRC Lab of Molecular Biology, where some of the first protein structures had been determined. And the person who was the director when I went there was Aaron Klug, who was I was doing some work in his area, and I had actually crystallized a protein that was of interest to him. So that was the sort of bait that I used to see if he would bite. And luckily, he didn't throw my letter in the garbage can. He sponsored me for a Guggenheim Fellowship, and I went there and learned crystallography. And when I came back, I collaborated with a colleague of mine, Steve White, and what we did was we took each protein from the ribosome, tried to crystallize it, and then solve its structure. So instead of balls, we had a detailed picture of each protein, but we had no idea where they were in the ribosome, how they fit into the ribosome. And of course, the 2/3 of the ribosome that was RNA was completely, a black box to us. So after a while, this sort of thing seemed like stamp collecting. We were getting nowhere with understanding how a ribosome would work. It would be a bit like going to an automobile parts store. You've never seen what a car looks like, and then just looking at different parts and saying, oh, isn't that interesting? But you have no idea what it does or where it fits in the car, okay? So what was the way forward? Well, the way forward was started when Ada Yonat, who's an Israeli scientist, went to Heinz Gunther Wittmann's lab in Berlin. He was a director of a very big lab in Berlin, and he was a very famous ribosomologist. And together, they obtained the first crystals of the large subunit of the ribosome. And this actually turned out to be a big feat because it was about almost an order of magnitude. It was about 5 times larger than the largest thing that had been crystallized up to that point, apart from very regular objects like viruses. So we had crystals of the large subunit, and then a group in Russia headed by Maria Garber used a different species to obtain crystals of both the small subunit and the entire ribosome. So now we had crystals of both subunits of the ribosome and the entire ribosome. But then the field was sort of stuck for many years. And so in the mid-1990s, even 15 years after the first crystals, there was no apparent progress towards a structure, not even a sort of what you call a fuzzy or low-resolution structure, okay? And Ada Yonat had spent many years to try and improve crystals of the large subunit. But even several years after those much improved crystals had been reported, there had been no progress towards, say, any kind of structure, not even an intermediate structure. And so after 2 meetings, when it really seemed as though the field was stuck, other groups decided that they probably needed to get in there and tackle the field and perhaps use different approaches. And so the Yale group decided, consisting of my mentor, Peter Moore, and Tom Stites, who had at one point offered me a postdoc, as I mentioned, decided to use the crystals that Ada Yonat had discovered in her lab and decided to see how far they could get with it. Now, I thought, well, that's fine. Let them fight it out with the 50S subunit. And I thought that it might be a better approach to try and improve one of the other things. There were no good crystals of the small subunit or of the entire ribosome. And so I thought, well, the whole ribosome was really too complicated and might come later. And so I thought, well, let's tackle the small subunit. It's small only in relative to the large subunit, but actually it was a huge problem. It's almost 1 million Daltons in molecular weight and had 150,000 atoms. So it was an enormously complex structure. And I had just moved to the University of Utah at that time. And I had two students, Bill Clemens and John McCutcheon, and they were both naive first-year graduate students. And I told them that, how about working on the structure of the 30S subunit of the ribosome? And since they didn't know anything about either ribosomes or crystallography, they thought it was a great idea, for a PhD project. And so we were off. And this guy, Brian Wimberly, was very, very knowledgeable about RNA structure and all sorts of things. He was a postdoc. He was actually on his second postdoc, and he knew too much. So he didn't want to start working on the project right away. But as soon as we had hints that it was going to take off, he was, luckily for me, he was happy to join the project. And this is me in my Bin Laden days. And you can see from this photograph that I'm the only guy who's wearing gloves when this picture was taken, showing that I was the only guy who was actually working when this picture was taken, okay? So we started off, we got good crystals, and then I went to a meeting in Sweden where the Yale group clearly showed that they were making progress on the 50S subunit. Ada Jonat showed data which she believed meant that the 50S subunit was impossible to solve. And she said, it's not worth tackling, it's an unfeasible problem. But then, which I was perfectly relaxed about, it had nothing to do with me. But then, to my surprise, she showed pictures of the small subunit, which now had improved diffraction. So instead of the Yale group and Ada duking it out on the 50S, the Yale group was now going to have a free ride, and I was going to be in a head-to-head competition that I had never wanted to with Ada Jonat. And she had, at the time, really 3 labs. She had a Max Planck outstation in Hamburg, she had a biochemical Max Planck support group in Berlin, and she had a lab in Weizmann with 15 or 20 people. And I had my 2 graduate students in Utah. And so on the plane ride home from Sweden, I almost thought I ought to give up. I mean, I thought, what is the point? And then I realized 2 things. One is I thought crystals of these things have been around for 15 years. Nobody's actually figured out how to crack the problem. And just because there's another crystal form now, it doesn't mean that you're suddenly going to, solve it. And so I thought, well, that's one thing. And the other thing I thought was, if I just stand back and watch other people do this, I'll be kicking myself for the rest of my life. This is the most important problem in my field, and it's a central problem in biology. And on the other hand, if I plunge ahead and give it my best shot, if it doesn't work out, I'll feel that I gave it my best shot. So I plunged ahead, and around this time, Harry Noller had personal reasons to visit Utah because he was in a relationship with a colleague of mine, Brenda Bass at the time. And at one of his visits, he said, I'm working on the crystal structure of the entire ribosome. And I thought, how could Harry, who's a biochemist who knows nothing about crystallography, be working on the structure of the entire ribosome? And I realized what had happened was he had hired this guy, Murat Yusupov. You might have recognized, he was a guy at the top right in that Russian group. And so Murat and his wife Gulnara were part of the original Russian group that had crystallized the whole ribosome. And he had hired Jamie Kate, who had solved one of the largest RNA structures at the time, which was the group 1 intron structure. So Harry had assembled a crack team with just the right kind of expertise. So suddenly, my quiet niche had become a kind of 4-way race. Now, the thing you don't do, what is the thing that you must not do in the middle of a tight race? And the thing you must not do is lose time. And what I instead did was to move to a different continent, okay, in the middle of a 4-way race. And so the question is, why did I do this? Well, you may remember I had spent a sabbatical year at the MRC lab of molecular biology, and I really liked the way the lab was run. and I thought it would be the ideal place to do this sort of work. In fact, there were two people I asked, and one of them for advice, and one of them actually was Steve Harrison, who introduced me. And the reason was I wanted to work on whole ribosome structure. And although the Yale group seemed to be making some progress, I had no idea how long it would take. And, university labs are funded on NIH grants, which last for 4 years or 5 years. And so at the end of year 3 or 4, if you haven't made your breakthrough and published a bunch of papers, they're not going to renew it. And if you just tell them, well, there's this group that's have had crystals for 15 years with a large number of people, but it's okay. I have really good ideas for how to get crystals and how to solve it. You can just sort of imagine what a panel how a panel might view that. I have a view. They would probably laugh and throw my application in the bin. And so I needed a place that had stable funding. And I also thought it would be good to go to a place which had really expert crystallographic colleagues, because this was a problem that was pushing the limits of the technology, of the technique, to beyond what had actually been done up to that point. And the LMB had a very collegial atmosphere, so people would be willing to help you and talk to you about your problems. And it's also an institution that had a history of supporting important problems for a prolonged period. It has had a series of Nobel Prizes, including the last 2 years. But if you look at the last 2 years, the prize was awarded for things that were started 25 or in one case, almost 40 years ago, okay? And so, it does have that sort of patient view of things. So I persuaded my very tolerant wife that we should take a 50% or so pay cut. It's actually 40%, but given the cost of living, it was more like 50% pay cut, and move to England away from our children who are now grown up. And luckily for me, she said yes, but she also said that it was the last time she was going to move. And she's sort of kept to her word ever since. Now, once we moved to Cambridge, it was not all smooth sailing. We'd made an initial breakthrough around the time we moved, but we ran into all sorts of technical difficulties, and before we arrived at something that was interpretable. And this is how Brian Wimberly spent his year and a half in Cambridge in front of a graphic screen with stereo glasses building the ribosome structure. Now, in order to get the data that we needed, we had to use these very powerful X-ray sources called synchrotrons. And you can see they're all over the place, from near New York to in the mountains of near the Alps in France to Chicago in the north of England. So I'd like to say, you can join a ribosome lab and see the world, but just like the Navy, said join the US Navy and see the world, I was lying too, because like the Navy, you're not seeing the world, you're seeing some inside of some horrible battleship. And they're not seeing, they're not going skiing or going to jazz concerts in Chicago or New York. They're actually seeing the inside of the synchrotrons and staying up day and night to collect data. Now, for the actual crucial experiment, we needed to align our crystals so that spots which were symmetric would show up on the same image. There's a technical reason for that I describe in the book. And there was only one beamline at the time that could do that sort of alignment, and also was bright enough in terms of X-rays to produce good data. And that was at the APS in Chicago. And I want to point this out. I've described this in the book because it shows you how chancy and how quirky science can be. So in October of 1999, I decided we had reached the limits of what we could do elsewhere, and I wrote to the people at the beamline in Chicago at the APS and said, can you give us beam time? And we had already published an intermediate version of our structure, so we had some credibility by the time. And I got a reply saying that, there's no time now, but there'd be time early the next year, and he would get back to me. Well, a month later, I repeated my request and didn't get a reply. Then a month later, I phoned the person and left a message on their voicemail, and there was still no reply. Then, by this time, the Yale group had cracked their structure. And in early January of 2000, Peter Moore wrote to me saying he was visiting England and could he give a talk. So I used the opportunity to ask Peter whether he would talk to his colleague Paul Sigler, who was a famous structural biologist, who was on the committee that oversaw this instrument in the synchrotron, and whether he could put in a word for us. So a day later, Peter wrote back saying that he's going to talk to Paul, which sounds sort of biblical, and actually, considering the effect it had on me, it really was. And then a day later, Peter wrote saying that Paul had contacted the people in the beamline and they would get back to me. And then a day later, the person at the beamline wrote back saying he was sorry, he had been really busy, but he was willing to give me time at the end of March. So this was 6 months after I had originally asked for beam time in what actually was a very tight race, okay? I mean, all the papers came out within weeks of each other. And, this really sad thing is 4 days after this intervention, Paul Sigler died of a heart attack on his way to work, okay? And this shows two things. In science, it helps if you know people who know people who know people, okay? If you're part of that inner circle, things can happen that, if I had been out of that inner circle, there'd be nobody who could intercede on my behalf. The second thing is this weird quirk, which is if I had waited 2 weeks, there would have been no Paul Sigler to intercede on my behalf. And I'm not sure what would have happened then. So, this just goes to show you how chancy science can be, how it can really depend on, the vagaries of fate. So anyway, we went off to Chicago, and over a 48-hour nonstop data collection period, we collected our data, and I talk about what that was like. And then what you end up is with a 3-dimensional image of the molecule if everything works. But this doesn't tell you what the structure is in chemical terms. It's just telling you what the 3-dimensional image is. You have to somehow interpret this 3-dimensional image in chemical terms to build a molecule. And it's a little bit like solving a large, complicated 3-dimensional jigsaw puzzle, but without an answer on the cover of the box, okay? And the way you would do that is you would do what you do for any other jigsaw puzzle, which is you'd look for areas that you recognize. And what you see is if you look over there, you can see that there are these ridges that seem to be connected with each other. And if you know what you're doing, then these ridges are essentially 2 strands of RNA that are interwoven around each other to form a double helix. RNA also forms double helixes like DNA. So suddenly, you've taken a piece of the image and you've built an atomic structure. You've determined an atomic structure. So now you know what a small piece of your molecule looks like in atomic terms, in chemical terms. And now you have to do that for the whole image. And so that's what you end up doing, and this involved spending months and months in those days in front of the graphics terminal, and you can see that you end up building the entire structure. And as we solved the structure of the small subunit, the Yale group solved the structure of the large subunit. And what these structures show is that the parts of the ribosome that stitch together the protein from the amino acids, or the part that actually reads the code, consist entirely of RNA, okay? And this helped solve a long-standing puzzle, which is if the ribosome is made of both RNA and proteins, but it's used to make proteins, where could the ribosome even have come from? And Francis Crick was the first to suggest that maybe early ribosomes consisted entirely of RNA. And then people like Harry Noller did experiments to suggest that that might be the case. But it wasn't until the structures came out that we really knew that really the ribosome is an RNA-based machine. Then a few years later, we saw the high-resolution structure of the entire ribosome with the genetic message and with the tRNAs bound. And this led to a number of things, including lots of snapshots of the ribosome in action to see how it worked, but also how antibiotics bind to the ribosome. And we saw structures of several antibiotics bound to the ribosome, and this is now leading to people trying to design better antibiotics, which have better properties and perhaps less toxicity and more effective against resistant bacteria. Now, some of the book also talks about the human side of science. And scientists are neither Spocks nor saints. We're driven by curiosity, by ambition, by a desire to succeed, to be first. We have egos, we have insecurities, we have anxiety, we have both rivalry and competition. We're often motivated by self-interest. We'll collaborate or compete depending on what's best for us. Sometimes competition is best, sometimes collaboration is best. But it's not all that sort of thing, because just as humans are complex with a mixture of altruism and competition and ego, that's the same is true in science. Many people helped me, they gave me advice, they provided reagents, technical help, and all of this was without any expectation. None of them wanted any money, they didn't want to be co-authors on my paper. It was simply to advance the cause of science. So you saw both the best and the worst of humanity in this microcosm of science. Now, thrown into the microcosm is the whole politics of recognition. And the corruption starts early with prizes. I talk about how prizes apply what I call a sports metaphor to science. In sports, if you have a 100-meter dash, the rules are very well defined, the goal is very well defined, and the measurement is very well defined. So you know who came first, second, and third. But that's not true in a scientific problem. Scientific problems are multidimensional. Different people contribute to different aspects of the problem. Even for the same sort of thing, it's often hard to know who made the really key, who had the key idea or the key experiment that advanced it. And unfortunately, we start this corruption very early. We start giving people prizes even in school, from undergraduate, graduate school. And then, we give young scientists early career awards. We pluck out people we think are stars and give them early career awards. Then, they start, wanting to get elected to academies, and then they hanker after major awards. And then it's complicated by the fact that many awards imitate the Nobel Prize in a slavish way by limiting it to 3 people. The Lasker Award, which is America's Nobel, used to have no such limit, and now it does, in a slavish imitation of the Nobel Prize. And that's very, in 1901, maybe it was reasonable. It's certainly not reasonable today when lots and lots of people contribute to to a particular problem. Another thing is the public regards the Nobel Prize as a sign of genius, okay? But the reality is more like how Shakespeare described it. Some are born great, some achieve greatness, and some have greatness thrust upon them. And the same is true of Nobel Prizes. People, stumble onto an important discovery, or maybe they're in the right place at the right time, or they have great colleagues who, give them advice, and they have resources. So there are all sorts of reasons, but it's given not for being a great scientist. It's given for making an important discovery or an advance. But because it's somehow, because of its history, it's been associated with greatness, people hanker after the Nobel Prize. And in my book, I somewhat scathingly refer to 2 diseases called pre-Nobelitis, which is when you sort of feel you're in contention for the Nobel Prize, and you get this anxiety every October and so on. And then there's also post-Nobelitis, people who get the prize for work they did 20 years earlier and are long past their peak. are suddenly thrust into the limelight and treated as geniuses, and they go swanning around the world, pontificating about all sorts of things, including the future of humanity and so on. But whatever it is, people sort of hanker after these things, and that warps their behavior. And I talk about the ribosome roadshow. There were several people, depending on, their own view of themselves, partly. Some of them I think were delusional. But anyway, there were, say, half a dozen people. And, so there are conferences all over the world. And, here we are, we've solved these structures and trotting around from one place to another, talking of our work. And so there's sort of this jockeying for three chairs, when the music stops. And it also almost takes on the kind of characteristics of a political campaign. And, suddenly I found the Swedes were terribly interested in ribosomes, okay? And they were keep inviting us to meetings in Sweden. And it's clear we were being auditioned. And at the last of these meetings, which was on the entire, what biologists call the central dogma, which is how DNA replicates itself and how it copies into RNA, and then how RNA is used to make proteins. So that entire field, people were invited to this meeting in Sweden. Now, there was a guy at that meeting who was the only Swedish ribosomologist to speak at the meeting. And he was sort of miffed that we had ignored his earlier work in the 1970s on one aspect of the ribosome. So at the banquet on the final night of the meeting, he accosted me and started chewing me out for ignoring his work. And at some point I got annoyed and started, arguing with him because actually I felt his early work was mistaken, okay? And the argument got so heated that a colleague of his had to come and separate us. And so I went home, and then 2 months later, I learned that he had been appointed to the Nobel Committee on Chemistry. So I decided that I was not going to accept any more meetings to Sweden, because why should I go there to be a supplicant and eat bad vegetarian food when I don't have a chance, okay? So I didn't go there. And then suddenly 5 years later, in one October, when I showed up in the lab with a flat tire and somewhat irritable, on my bicycle, that is, and I showed up somewhat irritable, and I got this phone call which said, this is an important call from the Swedish Academy of Sciences. And I didn't believe it. I told the guy, look, you have a good Swedish accent, but I'm not going to believe you until you put this guy Måns Ehrenberg on the line. And sure enough, Måns came on the line and said, Venki, I want to congratulate you, and I want to say you deserve it, but it's the last time I'm going to tell you that. So that was my thing. And so, given my track record, I didn't go to Harvard or Berkeley or Caltech or any of these places. I went to my local hometown university in India. I went to a state university in southeastern Ohio and wandered around from physics to biology using strange techniques. I never imagined I would be in this situation one December. And I just want to point out what is going on at this big table. This is if you look at just above my dot, that's the King of Sweden, who's at the center of the table. And this is Ada Jonat, and that's me. That is the Crown Princess Victoria, and this is Tom Steitz on the other side of her. And my wife Vera is busy in conversation with the German Vice Chancellor. Now, there are about 3 science prizes, or about 9 Nobel laureates that year, and 9 science laureates, in addition to 2 economists and a literature prize winner. But clearly the Swedish newspapers realized that the important thing that year was actually the ribosome, much more important than all those other prizes that they gave out that year. And why do I know that? Because the next morning, the thing that was on the front page of the Swedish newspapers was this, okay? Tom Steitz and me had made the front page of the Swedish newspapers. So clearly they thought that the ribosome was the important thing. And it had nothing to do with Crown Princess Victoria sitting between us. Okay, So in my last slide, I want to, looking back at my life, what did I do that somehow made it all work out? I could have fallen off the edge multiple times in my life, okay? And I always had a plan B and a plan C and so on. And one thing I did do was to keep my options open. So when physics didn't work out, I didn't sort of quit science or take some sort of, job that I didn't really want. What I did was to start over again in biology. Then when I had sort of a technique that really wasn't being very useful, I went on sabbatical to learn a new technique. And then I was willing to move places to always keep myself, give myself the best environment in which to do my research. The other thing that I would say I have is that I'm never afraid to ask for help. I'm not worried about seeming ignorant, things like that, because if you don't know something, the fastest way is to really ask for help from people who do know, and that involves talking to people. But you can't also talk to people all the time, because then you'll be influenced by what's fashionable and so on, and you won't really be able to do original work. The other thing you have to realize is everybody has imposter syndrome. You take a guy like me who went to sort of second-tier institutions, and then suddenly you find myself a postdoc at Yale or going to the MRC lab of molecular biology. Suddenly you find, my God, I've fooled these people, and in 2 weeks they're going to find out I'm a complete idiot, And everybody has this, unless they're sociopaths, okay? They probably don't have it, okay? And most people have learned how to cover up, and get along. And, sometimes if you're an outsider, you might feel it even more. And, if you're a minority, say you're a woman in a field dominated by men, you might feel, oh, maybe I don't really belong here. Or you're an outsider because of other reasons. You went to second-tier places, or you're geographically, from Asia or from the South or whatever, And you may think, really, I'm not like these other people, and maybe I don't belong here. And the thing is, you have to remember, everybody has this. And finally, science doesn't depend just on talent. It depends on lots of things. And it depends on money to be able to do the sort of work you want to do. It depends on skill, which depends on training, depends on patience, and it depends also on luck. And I'd like to stop here, but while we're taking questions, I want to just show you a movie of the ribosome in action, just to show you how amazing this molecule is and how complicated it does. So thank you very much. You can just watch the movie while you're asking.
Okay, I'm going to be running microphones through the audience. If you have a question, you can just raise your hand.
Here's the question. You think your odyssey would be possible in the age of technology?
I think so. I think even today, there are people changing fields. A lot of people are going in from mathematics into biology now because of the computational aspects. They're going in applying machine learning to biology. So I do think that, but it depends on having people, senior people, who are willing to be open-minded about accepting people from different backgrounds. I think that's what it takes.
One of the best aspects of your book is the extensive attention you give to students, both graduate students and postdocs. Is there any way to develop better ways to recognize these people? Because in all the groups that you mentioned, such junior scientists were very crucial to the ultimate success. And if I may, a quick second question. On several occasions in your book, which I bought at AAAS in February, you admit that you underestimated Ada-Ionat approach. And I don't understand why, because most of the time you were not, the Yale people had other set of problems with her, which I don't think we need to discuss here, but I don't see why you'd have a problem with underestimating her.
Well, the reason for underestimating her is because of 2 or 3 meetings in the mid-1990s, which suggested that she really, once she had gotten the crystals, she really did not have an idea of how to take the next step, okay? And that is the crucial time when 3 groups, the Yale group and we and Harry Noller, we were all at that meeting, where there were actually 2 meetings, one in 1995, which was a ribosome meeting, and one in 1996, which was a crystallography meeting. Those 2 meetings really gave us a feeling that her effort had completely stalled, and they did not have an idea of how to proceed to the next step. And so we went ahead, and then we started making progress. And in 1999, at a meeting in Denmark, it was clear that the other groups had all made their breakthroughs, but it was not yet clear that Ada Jonat had made her breakthrough. Her talk was really, if you ask most ribosome people, they were simply not convinced by anything that she showed there. And therefore, when only 6 to 8 months later, I attended a meeting in Grenoble and saw that she had now obtained rather good maps, not good enough to solve the structure, but still much better than what she had. That is when I say in the book that I had underestimated her, okay?
I have a question over here in the back. What are you working on now, and how do you support young scientists, like in your lab and maybe other young scientists that you're mentoring?
Sorry, how do I what? The second?
What are you working on now? What are your current research projects?
But what's the second part of the
And how do you support young scientists?
Yeah,
Thank
so what I'm working on, so in science, when you solve a problem, it simply raises the next level of questions. So now that we know what ribosomes look like, we're trying to understand how they work. Now, we've done a lot of work on how ribosomes actually do their thing, but now we want to understand, how does the cell regulate ribosomes? How does the cell decide when ribosomes are stuck and need to be rescued? How do viruses hijack ribosomes to do their own, make their own proteins? How do the ribosomes of mitochondria, which are these organelles inside us, how do they function? So there are all sorts of different questions that we're trying to answer, and there are more powerful techniques now which are which involve cryo-electron microscopy. So today, nobody would go through all this odyssey that I described. You would simply use electron microscopy to solve structures. So that's the first part. The other part is, how do you support students? Well, I never tell students in my lab what they should work on. Rather, they come to my lab and I say, look, these are the open questions in the field of translation, which is how proteins are made. And why don't you think about it and pick something that you think is workable, and then, or come up with a couple of ideas. And here are some things you might think about. And then they choose the thing. And when they choose it, they feel it's their problem and they feel invested in it. And because, science is hard, a lot of times most of the time things are not working. And they have to feel motivated to come in every day and do their thing.
Hi. He's here. He's a caller. Thank you for the talk. I have a question you mentioned about sometimes it's very difficult to identify who made the key concept or key contributions. And how do you fix it? For example, go back to the 50s when James Watson and Francis Crick discovered DNA. Graduate students and a postdoc can publish a paper in Nature. But now it is impossible for graduate students and a postdoc.
Yes.
And how do you fix this? How do I identify who made the key contributions?
I have to say, one thing is that of all the prizes, oddly enough, the Nobel Prize is one of the few big prizes that recognizes actual contributions by students. For example, the telomerase prize went to Carol Greider. She shared the prize for work that she did as a graduate student with Liz Blackburn. Or George Kohler was a postdoc with Cesar Milstein and shared the Nobel Prize for antibodies. But in general, it is a big problem, okay? And part of it is that I think the view is that, the students and postdocs are there to be trained, but really it's the lab head who's setting the agenda, who has the vision and is really driving the problem forward. And so that is a particular view. It's not always true, okay? And it's certainly true that the lab head would not be able to do it with students, without students or postdocs. It's very, very rare, maybe in theoretical physics or something, but in experimental biology, that's very, very rare. However, there might be a view that, well, if it hadn't been for this student or that student, some other student might have come in and done the same sort of work. So that's the kind of view that the prize juries take. But as I point out in the book, I do actually point out this particular problem, which is that it doesn't recognize, that science is done, by teams and not by necessarily by individuals.
And we just have time for a couple more questions.
Hi. So I'm a freshman, first year undergrad right now. Here. I just want to ask if you have one piece of advice for, early stage students, like undergrad students who are looking to maybe go into like biochemistry or cell biology in the future, in the next, 10, 20 years.
So, sorry, what did you want?
So if you have any advice for like, so I'm a first year undergrad right now. So if you have any advice for like younger students who want to go into like biochemistry or cell biology in the next.
So the one thing I would say is try to see what it is that fascinates you and interests you, and don't worry too much about fads, okay? If you're a first year undergrad, did you say you're an undergrad? Okay, you're first year undergrad, you have 3 more years of undergrad, then you've got maybe 4 or 5 years of graduate school, then 3 or 4 years of postdoc. We're talking 10, 15 years down the line before you start work. So whatever you think is really fashionable now may not be fashionable in 10 or 15 years. For various reasons, people might realize it's not that important, or it might actually be finished, and the interesting problems might be solved. So I would say just stay interested and stay open at each stage of your life, okay? As an undergrad, you should be sampling very widely, and I would say you should acquire a very good background in what I call the 3 or 4 core disciplines. Mathematics, which underpins all of science, physics, which also underpins, a lot of science, and chemistry, and then biology. So I would say if you train well in those 3 or 4 disciplines, that gives you the tools later to enter almost any area of science.
Hi, I'm over here. I have a question. So you said that you were in a competition with the Yale team. So did you guys ever collaborate with each other or like bounce ideas off of each other?
No, so we had a question before about, competition, and especially with Ada. And I think, part of it was that, Ada had the field to herself for a long time. And so she was not entirely happy with all these other people coming in to what she sort of felt was her domain, okay? So relations were not great. And with the Yale group and us, we were friendly because one of them was my former mentor, Peter Moore, so we remained on friendly terms, but we weren't quite so direct competitors because they were working on the other subunit. But nevertheless, because it was important that, we get there first. We weren't all that communicative either, okay? We were not exactly, open about it. And that is a problem with this sort of, not all of science is a race. Most of science isn't like a race where the goal is very well defined, and it's a question of who gets there first. A lot of science is very open-ended and exploratory. So you mustn't get the idea that it's always like this, but often it's like that, I mean, once a problem becomes known to be important, then it becomes a goal, and then it becomes like that.
Last question in this top corner, right up here. Hi.
Hi. Was the concern about new antibiotics something that you were working with even as a younger scientist, its relationship to the ribosome, or is that a new concern, the relationship of
Well, the
structure of
antibiotics were very useful because you could write on NIH grants that lots of antibiotics bind to the ribosome, and therefore, if you understand the ribosome, you might one day help get better antibiotics. And for 20 years, we wrote this without believing it. But it turned out to be true, okay? By accident, we realized we were actually telling the truth. And so now we do know how antibiotics bind. But that's not what motivated us initially, okay? What motivated us was a central problem in biology, okay?
Now with the advancements and the creation of supercomputers, do these tools change the way biologists do their research?
I think the advances in computing have always gone hand in hand with advances in structural biology. So from the very first protein structures, they used the state-of-the-art computing of their time, which was an EDSAC or something like that, which took up an entire room. And as the problems in biology kept getting more complex, the computing became more powerful. So we're always sort of ending up using the most powerful computers that there are.
And I think that's all the time we have for this evening. One last round of applause.